Yay. All right. Woo. We are here. Good morning, guys. I am, let me turn this off. Good morning. Yes. Yes, everyone. Thank you. Thank you so much. Yes, it's official. It's official. I finally am a doctor. I am now Dr. Panko. See, I even changed the PowerPoint slide because I could. uh, but man, so I've been so focused on so I don't know how familiar you are with how this whole process goes, but um, I've been so focused on the presentation aspect of this of becoming a doctor, then I didn't even think about the fact that I would have to do a bunch of revisions. So so I have until today to finish revising my 138-page document of my dissertation. So yeah, so even though I'm done and I am a doctor, I'm not getting any sleep, as you can probably tell. But that's okay. I'm still thank you, Manuel. I'm still just ridiculously it still doesn't feel real, I'll be honest, especially given this whole, situation. So but yes, so it is recorded. If you I am gonna post it on YouTube once the recording is finished being processed. There's aspects of the recording we're not allowed to share to the public because like the private deliberations and stuff. So, but anyways, yes, thank you guys. I told you at the beginning of the semester it was going to happen. Are you done after this? Here, people are writing stuff. I'm not even reading it here. So everyone's saying congratulations, and Gabrielle's asking, are you done after this? Any big moves? LOL. So yeah, actually, I do. I have a job lined up at Michigan State University. To start later this year as a postdoc. So I have a postdoc position at the Department of Plant Biology at Michigan State University. And I'll be working with the amazing Dr. Emily Josephs. She is new faculty there. I went out and met her last November. We met at the Evolution Conference in last year, and then I went to go visit her last November, and she and I have almost identical research interests, and we get along very well. So I'm excited about that. It's going to be a big change, though. I've been living in Brooklyn, New York for 16 years, so it'll be interesting to move out to Michigan. But I like a lot of water sports and I like hiking and things like that. So I'm excited for the change. It'll be a short-term position, only a year or so, year or 2 years, something like that. Yeah, so I'm excited. I'm excited, but also nervous. A lot of things changing all at once, but change is good. Change is good, and we just gotta roll with the punches. But no matter what you guys end up doing down the road, I'm here for you. So if you need help ever with like grad school prep, or if you're applying to medical school, you need a letter of recommendation. I'm here for you guys. So if there's anything I can do to help you on your journey, let me know. Okay. I'm here for you guys. I know you guys have big dreams too. So, and you're going to get there. You're going to get to your big dreams. Do it. Speaking of that, we got to go though. This is our last lecture. Sad face, right? Last lecture. And I'm going to do my best. Okay. I'm going to do my best to get through all of the material. There might be a chance it run over just a little bit because, but I know I miss this class too. Maybe we'll figure out something. I don't know. Once I finish the revisions and things calm down a little bit, maybe I can think of some fun way for those of you who want to meet not at 10 a.m. But maybe we can do some other fun plant thing or something off the record. Just we'll think if we can do something fun like that. Okay, so let's just dig into it. Let me drink coffee before I start. Also, I don't know if you can hear, my heat hasn't like turned on for like a month, and then the day I gave my presentation in here and since then it's just been going nonstop. So it's like 90 degrees in this room right now, and if you hear a hissing noise, like if you hear that, that's unfortunately my heat. I have no control over it. The knob is broken, so I cannot turn it off. So sorry about that. But at least it's probably not 90 degrees wherever You are. Phenology, okay, yes, phenology project. Here's what I'll say for phenology project. Just send me whatever you have. Just send me whatever you have. Use the directions I gave you, right? PDF and a Excel, but just send me whatever you have. And obviously, I wasn't expecting us to be quarantined when I created that assignment, but it will be fun for me to look through your pictures, the few, even if it's just a few pictures from a week or 2 or 3 weeks or however many. Just send me whatever you have, and then at least I can, see your lovely faces and look at what trees and stuff you found around. So yeah, just send me whatever you have. Don't sweat it. Don't sweat it. All right, let's get started. So yes, instructor Dr. Rebecca Pinko. Boom! Oh man, took 6 years. Took 6 years to be here, but sorry, couldn't help it. Boom! It still doesn't feel real. Totally doesn't feel real. But it's true. Okay. All right. Yeah. Woo! So let's go. We're gonna finish the Angiosperms today. I'm looking at the clock. We're gonna do the best we can. Particularly Megasporogenesis, Microsporogenesis. We already know there's a lot of flowering plants, right? We already know there's a ton of diversity in form. That's part of the reason why so many people like working with them. They're just incredible to work with because of the diversity in flower shape, but habit and habitat as well. They're just a fun, charismatic group. Lots and lots and lots and lots of plants to study. And you may remember. So first thing we're going to talk about is flower arrangements. So flower arrangements, and these are not like arrangements like you get like my husband got me these, like, see, I have roses here. I'm not talking about like a flower arrangement, right? I'm talking about how we describe how flowers are arranged on a plant. That's what we're talking about. And so you might remember we talked about this before, that a flower is actually a specialized shoot system, right? So a flower, if anything, if you want to make a comparison, a flower is kind of like a strobilus in the sense that it's a specialized shoot system. You have a stem and you have basically modified leaves, right? So like petals are modified leaves, stamens are modified leaves, a stigma is even a modified leaf. So you can think of it as a specialized shoot system. And the flowers bear the reproductive structures, right? Just like when we talked about strobilus in the past, the strobilus also bore reproductive structures. However, in the flowers, the male reproductive parts are now called, let me close this so I can see, there we go. Male reproductive parts are now called the stamens, as I said, or the androcium. Female reproductive parts are called the pistils and carpels, or the gynosium, right? I like stamens and pistil, but you can use the more advanced term if you prefer. All right, come on. So this is about flower arrangements. So flowers can be solitary, or what we say born singly. You might remember when we were talking about leaves on a strobilus in the past. We said sometimes the strobilus leaves could be born singly, or sorry, no. We were talking about sporophylls in the past because we said sometimes that they could be born singly or arranged in a strobilus. Well, flowers can be born singly also, meaning they're by themselves. So like a rose, example of a flower that's born singly or solitary. It means that there's a stem and then there's one flower on top. But you can also have flowers grouped together, and when flowers are grouped together, we call this an inflorescence. So here we have an example from this lily agapanthus. You can see there's lots and lots of flowers there, but they're all localized in one area, and we call that an inflorescence. Okay, so the stalk, the stem part that's bearing a solitary flower is called a pedicel. But if you are a stalk bearing an inflorescence, we call it a peduncle, just like a term we have seen in the past, right? We saw peduncle before, back in the strobilus days. And so it goes further than that, right? You have not only do you have solitary flowers and inflorescences, but then you also have different types of inflorescences that you can describe in different ways, depending on how the many flowers are arranged. So here is a group. This is like a summary slide of all the different inflorescent types that we talk about in this class. But we'll go through each of these individually. I just wanted to get you kind of understanding how these little pink drawings work, where every circle in these pink drawings indicates a single flower of an inflorescence. So this is a panicle. Hydrangea is an example. There's certain types of hydrangea and viburnum that have this flower shape, this inflorescence shape. Just still okay to say flower shape, where they're kind of loosely clustered, but they're not like opposite each other, just kind of like loosely clustered on a stalk. Then you have spike. So here, this is actually a picture of lupine. You remember from the first documentary assignment we have where they were talking about lupine. This is lupine. Lupine is an example of a plant that has a spike. And remember, in these pink drawings over here, each of these little circles indicates a flower. Then you have a raceme, which is similar to a panicle, except that, see, a panicle has many things coming off of the single stalk, whereas a raceme only has one flower coming off of each of the little stalks. Then you have, this thing is called a quorum. The thing that's, and this is a picture of yarrow, the plant yarrow. Quorum is kind of shaped like a triangle. That's how I think about it. Each of the stalks underneath the flowers, here, I'll annotate. Each of the stalks, so like the first one get shorter and shorter as you move up the stem, so that the flowers are all at the same level at the very top to kind of create like a landing platform for pollinators. Okay. Oh boy. Then you have a simple umbel. An umbel is kind of like a quorum, except that at the umbel, all of the stalks start at the same location and they're all the same length. So learning these different terms can be useful if you're ever in the field or on a hike or something, or even walking around your neighborhood and you see, let's say you see a flower that you think is just so beautiful and you want to know what it is and so you could go plant it in your garden or give it to somebody as a gift or something like that. But you might have no idea what kind of flower it is, right? If you have terms like this, if you can like look at the plant and see how the flower is arranged, then you can go to a book or even online and it gives you clues as to how to find out what kind of plant it's from. So you have umbel, which is where all the stalks are the same length, but they still kind of the flowers all are at the same distance from the top of the stem. And then you have a compound umbel, which is like an umbel of umbels. So like here's a simple umbel. And then on the compound umbel, each part of the umbel will have an umbel, right? It's an umbel of umbels. So here's an umbel, and then each end of the umbel has an umbel. So I always say it's an umbel of umbels. Queen Anne's lace and a bunch of things in the parsley family. So celery, dill, things like that have umbels or compound umbels. Carrot, yep, that's in the parsley family. It's true. Although I don't know if I've ever seen a carrot flower. I have to think about that. I don't think I ever have seen a carrot flower. Now, when I'm done lecturing today, I'm gonna go Google what carrot flowers look like. Oh, it's similar. Yeah, it makes sense if it's in the parsley family. Okay, so compound umbel. Then you have catkins. Catkins are interesting. They don't really look like a normal flower, as it were. So right now actually is a good time of year to see catkins. Birch trees are an example of a plant that has catkins. Catkins, to the naked eye, if you didn't know better, kind of look like a strobilus. You didn't know better and you just saw that, you might say, oh, well, that looks like a strobilus to me. And you wouldn't be too far off because it kind of does look like that. But they're not, because they're from angiosperms. So they're types of flowers. this flower, maybe some of you have seen before, right? Sunflower. Sunflower is an example of a inflorescence type. So you might have looked at a sunflower in the past and thought that that was just one flower, right? Sunflower, one flower. But actually, a sunflower should be called sun inflorescence because it's actually a type of flower called a composite flower, which is composed of many flowers, many. So if you look at the schematic on the left, right, you see it says composite flower. It's an inflorescence, and it's composed of actually, not only is it composed of many flowers, it's also composed of two flower types. One is the disc flowers, which are the part in the middle. And so here, I'm going to get out of red. I'll go to blue. So you have disc flowers. These flowers all here are the disc flowers, all of these little guys. And then you have the ray flowers. Yeah, so it's actually, like I said, it should be called a sun inflorescence, not just simply a flower, sun inflorescence. Here is a drawing of that so you can see it better. So this is a cross section of a sunflower from the side. And you can see the disc flowers are all in toward the center. You see these little kind of things that are coming off like this. On the top of each of those flowers, those are the stigmas of the disc flower. So we know that the disc flowers bear at least female reproductive structures, and that would make sense too, because when the sunflower gets pollinated, all of the sunflower seeds that we like to eat will be located on the disc. Okay, so angiosperms, like all plants we've learned about for the most part, can be monoecious or dioecious, right? And if you're monoecious, you can have either perfect or imperfect flowers. Because remember, the terms monoecious and dioecious do not refer to a flower, they refer to a plant individual. So if you're a monoecious plant individual, it means that on your plant body, you have male and female reproductive structures on your plant body in one house, the house being your plant body, right? So if you're a monoecious plant, you can have the male and female reproductive structures together in a perfect flower, or you can have both types of flowers, male and female flowers, on your plant body, and you're still a monoecious plant because you have both male and female reproductive structures on your plant body. They just happen to be in imperfect flowers with either male or female reproductive structures. However, if you're a dioecious plant, right? Dioecious meaning only you have things in two houses, right? Two plant individuals. Because of that, if you're a dioecious plant, you can only have imperfect flowers, right? There's no other way around it. You have to have imperfect flowers if you're a dioecious plant. Oh, yeah. And then this is just a recall of saying, again, that the male reproductive structures are on the stamens, which are made up of the anther and the filament. Female reproductive structures of a flower are the pistils and carpels, which are made up of the stigma, style, and ovary. So now we're going to talk a little bit about angiosperm reproduction. So as I mentioned, so perfect flowers versus imperfect flowers. So if you look on the left here, this is an example of a perfect flower. We can see with our eye, like in the middle here, each of these little things coming up in the middle are the tops of stigmas. Or sorry, yeah, are the stigmas of the female reproductive structure. And all these little yellow dudes are stamens. So this is a perfect flower. It bears both male and female reproductive parts on the same flower. Perfect flower. And then on the right, you have imperfect flowers. Each flower either bears male or female reproductive structures. So this is our catkin from before. I don't know if you can see this thing hanging down. Is that catkin flower we just saw? And catkins tend to be composed of only male flowers, which and they tend to be wind dispersed. The pollen tends to be wind dispersed in catkins. The reason I mention that is because remember I said the catkin kind of looks like a strobolus, and the strobolus of like a strobolus of a gymnosperm, and those are also wind. The pollen is also vectored by wind. That's why I mentioned that. But anyways, you don't have to have a catkin to be imperfect. There's a whole bunch of different examples of imperfect flowers. The point here is just that if you're in an imperfect flower, you have either male or female reproductive structures on your flower. Okay. Oops. Go forward. Right. So we're going to go through angiosperm reproduction. Yep. Sorry, my clock is hidden by this thing, so I keep looking to see how I'm doing on time. Actually, let me try to move it a little. Yeah, all right. Well, I'll still have to look to the side, but at least I'll be able to see it. Yeah, so we're going to talk about angiosperm reproduction, and for the sake of simplicity, we're going to go through it using a perfect flower. All right, so here's a perfect flower, and we have in the boxes the different parts, right? The male and the female reproductive structure. So on the left here in this box, we have the female reproductive structures, which is the pistil or carpels, which is composed of the ovary, style, and a stigma. In this box here on the other part is the male reproductive structures, which are the stamens, which are made up of an anther and a filament. So this is, let me move this. So we'll go through the different parts again. So this is the stigma. This is the pistil, right? Or gynoecium, but I like pistil. And it's made up of the stigma. Come on, computer. It's made up of the stigma. So this is just that same thing, but bigger, right? So it's made up of the stigma. Remember, the stigma is sticky stigma. And its job is to catch pollen. Oops. Oh yeah. And stigmas so even though a lot of times you see these diagrams and the stigma is just like this little, it's nothing special. But stigmas can actually look very different depending on what species you're looking at. So this is a diagram here from an older publication, and each of these is actually an example of a stigma. So, I mean, look at D. Look at all this. Look at all what D's got going on. Look at E. E doesn't even look like a stigma. E looks like a peacock feather or something. And then look at F. F looks like a Muppet or something. And then I's got these little like curly Q things. So there can be a lot of diversity in stigma shape. Here, instead of a really old diagram, check these pictures out, which shows you the different other examples of what stigmas can look like. So very, very pretty very, very pretty, but always very, very different looking depending on what species you're talking about. Look at this pom-pom guy at the bottom here. Really, really cool. So this stigma on the bottom has actually like 5 tips, all that connect to the same style. The one on the left and the bottom has 2 tips. Yep. So a lot of diversity in stigma shape. So stigma is sticky and it catches the pollen. That's its job. The style is the long thing, usually long thing, that holds the stigma away from the ovary. And it's solid, right? Not hollow, even though it kind of is in the shape of a tube. And then the ovary holds one or more, ovules, right? So the ovules are now inside of an ovary in the angiosperms, right? Because they're not naked ovules anymore. Now they are in an ovary. And as we've already talked about, right, one of these kind of like core concepts of the course is the fact that as we go through the plant kingdom, right, the gametophyte parts get smaller and smaller and smaller and result in this better protection of the female reproductive cells, right? So angiosperm ovules are much better protected than the gymnosperms. We saw this actually before. But the way they're better protected is, one, the ovules are inside of an ovary, and two, each ovule has two integuments, right? So in this schematic here on the right side, the blue thing is the ovule, and the pink thing is the ovary. But don't get confused, right? You have often many ovules inside of a single ovary, but this schematic here just simplifies it to show you that to compare it with a gymnosperm ovule, which is naked. Okay, so let's talk more about that ovule, right? So inside of the ovule, so let's take a step back. Inside of this ovule, right? There, I'll draw. Here's your ovule. Here's your angiosperm ovule, right? And inside of that angiosperm ovule is this megasporocyte, right? Which is going to undergo meiosis to produce, to ultimately produce the female gametophyte, right? Okay. So this is a ovule on the right, showing you the parts of the reduced female gametophyte plant. So the female gametophyte plant now is so reduced that it only is made up of 8 cells now, or rather 8 nuclei. Well, a mixture of cells and nuclei, but 8 components, 8 things, 8 cells basically is what now makes up the female gametophyte. That's like so reduced from when we were talking about the gametophytes back in the non-vascular plants, right? Where you had a whole living plant. Now we're just talking 8 components, 8 cellular components. And the parts of the female gametophyte plant are these 3 antipodal cells, which you can see here, 3 antipodal cells, 2 polar nuclei, 2 synergid cells, and an egg cell. Here where it says micropylar N, that's just showing you where the micropyle is to get you oriented. Don't worry about the other end. And this is one thing I never really liked about botany, but unfortunately the female gametophyte is in the angiosperms is also called the embryo sac. Ugh, never liked that. Embryo sac. Okay. Okay, so you don't just go from having a megasporocyte to whammo, right? A female gametophyte plant. There are steps. We're going to talk about them right now. And the steps of going from a megasporocyte to a female gametophyte plant is called Megasporogenesis, right? Remember, break the term down, right? Mega in plants refers to female because the female structures tend to be larger. Sporo refers to spores, right? And then genesis is the creation. So it's the word Megasporogenesis is the creation of the female spores, right? In this case, the functional megaspore. So what happens is, here, I have it written here. So Megasporogenesis is the process of producing one functional megaspore, which then gives rise to the female gametophyte plant bearing an egg. And so here, this is one of those schematics similar to what we saw when we first were learning about meiosis and mitosis, right? So you have a developing ovule or 2N ovule. Inside of that is that megasporocyte, which is also 2N. That megasporocyte is going to undergo meiosis 1 and 2 and cytokinesis to make that functional megaspore, which is now 1N because it went through meiosis and cytokinesis. The functional megaspore is going to go through mitosis, 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 3 rounds of mitosis, and then cytokinesis. That's why when plants, you never assume cytokinesis, because you have these times where you have many mitotic events before a cytokinetic event. And that results in the mature ovule, which is now, and that results in the mature ovule, because remember, the ovule is the outside, right? The ovule is the outside. The ovule is part of the sporophyte plant, but inside of that ovule is the female gametophyte plant, which is one end with her egg. So here on the left is a cross section through a lily ovary, and you can see inside of the ovary are many, many ovules, right? Too many for us to even count, and we can't even see some because they're behind the middle part. So we're going to talk about this process inside of one of these ovules. And we're going to go through the steps. It's the steps I just showed you on the previous slide, but now we can see how it works. So you start with this developing ovule, and ultimately what you're going to get to through all these steps is the mature ovule, right? That's the same picture from the previous slide. And inside of the mature ovule, after all the steps are done, you'll have that female gametophyte plant with the egg. So how do we get there? So here's the steps. Here's the steps of Megasporogenesis. So you have inside of a developing ovule, the little letters in this, see like I and M and C, it tells you in the written part what those stand for, right? So little I stands for integument, little M stands for Megasporangium, little C stands for spore mother cell, right? Or megasporocyte. So in the developing ovule, you have these 3 things. And the megasporocyte is going to undergo meiosis and cytokinesis. So you can see in there, now we have a tetrad of spores, right? But they're stacked vertically in this diagram. So see, S are the spores, right? So 1, 2, 3, 4 tetrad of spores, the result of meiosis and cytokinesis. And so just like we're used to before, except now that it's happening inside of the ovule. Notice now you have 2 integuments, right? So you started off with 1 as the ovule was developing, but then eventually a second one will grow to start to surround the ovule. 3 of those megaspores will abort, and only 1 of those megaspores will become the functional megaspore, right? That functional megaspore is going to undergo mitosis event number 1, because we're going to have 3 events of mitosis here, to produce 2 daughter nuclei. Then there's going to be another mitosis event, so that now we have a total of 4 nuclei. So basically each of those nuclei are going to undergo mitosis, and now we have a total of 4 nuclei. You see them all floating around in there. Then we have a 3rd mitosis event, such that now we have 8. So each of those four are also going to undergo mitosis, so that now we have a total of 8 nuclei. And then finally cytokinesis occurs after those 3 mitotic events. And now, so we had what? 8 daughter nuclei because of 3 mitotic events. And each of those nuclei are going to be components of the female gametophyte plant. So now we have the female gametophyte plant with 8 components, right? And those 8 components, so each of those nuclei, one of them will become the egg cell. 2 of those nuclei will become the synergid cells. 2 of those nuclei will become these polar nuclei. And 3 of those nuclei from the previous slide will become what are called the antipodal cells. Let's see, I think I put colors here to help you with this. I do, right? So here, when I post the slides, you'll have this slide both with, without the color, so you can actually see the diagram. But then I also do color code it for you guys. So now we have a mature ovule. So here's the mature ovule. And the components of that mature ovule, which we've said on the previous slides, are 2 integuments that are surrounding the megasporangium. Inside of that megasporangium is the megaspore, right, which has a megaspore wall. And inside that megaspore, right, inside that megaspore wall, so inside the megaspore wall, you would have had the functional megaspore. But the functional megaspore just went through all those things we showed on the previous slides. So now inside of that megaspore wall is that female gametophyte, this, right, which has an egg, right? I know it might seem a little complicated. I recommend that you, once this lecture is over, you just go through the slides and you can listen to what I said. I'm saying the same thing over and over. It's just I'm showing it to you in different ways so that maybe it makes more sense. Okay, but the point is now the egg is very well protected, much more protected than it was in all the other plants we saw in the plant kingdom. All right, so that's Megasporogenesis, which is a little complicated because of all these mitotic events, but it's okay. We're smart. We can handle it. Now we're going to talk about the male parts of the, the male parts of the plant, right? The stamens. So the stamens there they are. They're highlighted there in red. Have anthers, which produce pollen. And they have a stigma, They have anthers that produce pollen. And the stamens have a filament that holds the anther out of the flower. And we saw in the gymnosperms, right, we became familiar with the pollen grain, right, in the gymnosperm. So angiosperms still have pollen grain. It's just that now the male gametophyte in that is represented by that pollen grain is even more reduced than even in the gymnosperms. So now it contains, it's made up of the pollen tube cell, which we are used to, and the generative cell. That's it. No more prothallal cells. Because it's like, what were they even doing in there in the first place? And so by the time you get to angiosperms, it's like, well, these aren't serving any function. So why do we even have these? So we just move on and have only pollen tube cell, which grows a pollen tube, and then the generative cell, which generates the 2 sperm. So further reduction of the male gametophyte plant in the angiosperms. And you have similar, although way less complicated process to make the male gametophyte plant. And it's called Microsporogenesis. Remember, Genesis is the creation of, or the beginning of. Spore refers to a spore, and then micro in the plant world usually refers to male structures because they tend to be smaller. So therefore, microsporogenesis is the process of creating or generating microspores, which ultimately give rise to pollen grains, and the pollen grains contain the male gametophyte, which is made up of now just a pollen tube cell and a generative cell. So here's a similar overview of like what we saw in the previous, right? You have a microsporangium, or a pollen sac, same thing, which is 2N. Inside of that is microsporocytes, which are also 2N. They're going to undergo meiosis and cytokinesis to create microspores. Each of those microspores is going to undergo mitosis and cytokinesis, and the result will be a pollen grain, which has a pollen tube cell and a generative cell. Oh my God, the heat! I wish I could turn it off. So hot in here. If you join late, if you hear a hissing noise, it's my radiator. I can't turn it off. Although I really want to. So let's look at microsporogenesis. Like I said, it's not as complicated as megasporogenesis. So here on the left, we see anthers that are red on filaments. If we were to take one of those anthers off, do a cross section through it, we would see that each anther is composed of different Microsporangia. So in the case of this anther, when we cut it up, this anther has 4 microsporangia. These are them, but honestly, 1 microsporangia is like, so each of these things, each of these banana-shaped things, whoops, okay, are each a microsporangia. But then we're gonna, on the next slide, if you do a cross-section through it, we're gonna look at it. It's gonna look like this butterfly shape that we see here, and that's what we'll see on the next slide. So here's that butterfly shape. This is a cross-section through an anther, showing you microsporangia. So here is, yep. So look, anther in cross-section, 4 microsporangia shown. So just like on the previous slide, I'll draw so you see. So here, this is 1 microsporangium. This is 1 microsporangium, right? And then there's 2 more, right? Inside of each microsporangium are microsporocytes. Whoa, still not good at that. Inside each of those microsporangia are microsporocytes, right? That are going to ultimately go through meiosis and cytokinesis to produce the microspores. Oh, see? I knew I couldn't draw well. I did it for myself, right? Microsporangia, 4 of them. 4 microsporangia. The microsporangia have that inside, like I said, have the microsporocytes. Each microsporocyte is going to undergo meiosis and cytokinesis to make a tetrad of microspores. Let's talk about one of those microspores. The microspore is going to undergo mitosis and cytokinesis to give rise to the male gametophyte, which is now what we call the pollen grain. However, we're in the angiosperm, so it's even more reduced than we saw previously in the gymnosperms. And the pollen grain now consists of just a pollen tube cell and a generative cell. So once the pollen grains are made, then the microsporangia, aka pollen sacs on the anther, open and release pollen grains. So remember, the anther is not releasing microspores. The anther has microspores inside that turn into pollen grains, and it's pollen grains that are released from the anther. So here's that same anther in cross-section. Now it contains pollen grains instead of microsporocytes or microspores. Now it's pollen grains, and it's ready to open, and it opens and releases the pollen grains. All right, so now we know how pollen grains are made, and we know how ovules with the female gametophyte plant are made. So now let's look at how reproduction occurs in angiosperms. So the pollen grain just left the anther, right? Pollen grain just left. There it is, flying away from the anther. See, there's a stamen, it's showing you the anther, and there's that pollen grain flying away. The pollen grain is going to be brought to the top of a stigma via what we call an agent of pollination. Ooh, sounds so like official, right? Like James Bond, like it's an agent of pollination, but yeah, that's actually what we call it. So an agent of pollination is going to transport a pollen grain to the top of that sticky stigma. And when that occurs, when pollen grain lands on sticky stigma, that's what we call pollination. Pollination, there you go, that's pollination. So there's each of those yellow dots are pollen grains, and they have landed on top of this stigma, which in this case has 5 branches. Agent of pollination, what is that? Okay, so agent of pollination is just whatever way the pollen grain travels from the anther to the stigma. So you can have abiotic agents of pollination, which is water and wind. Oh, thank God. Whew. Finally turned off. And you can have biotic agents of pollination, such as insects, birds, reptiles, small mammals, and small mammals, even including bats. So these are examples of abiotic agents of pollination. So wind on the left, water on the right. So that's pollen floating through the wind or pollen floating on top of water to go to plants. And then these are examples of biotic agents of pollination, right? So you have insects, like here, bees, wasps, butterflies, moths. You can even have pollination by beetles. You can even have pollination by flies. Actually, a lot of pollination occurs by flies, but we don't even talk about them. We don't give them any credit. But you can see here with this big giant flower, you have a fly that's going to be the agent of pollination. You can have birds as agents of pollination. You could have reptiles as agents of pollination. And small mammals. I love this slide. I love this slide so much. I mean, look at this guy. Look at this guy. Oh man, look at these guys. Aw, love this slide. I study plants, but I still love cute pictures of animals. Super cute. Super, super cute. Yeah, so you can have small mammals. Yeah, they're having a party as agents of pollination. All right, so pollination occurred, right? Pollen landed on the top of a stigma, right? Okay, so that occurred. Now, what's going to happen? Well, the pollen grain lands on the top of the stigma, and the pollen tube cell is going to grow, right? There you go. So pollen grain lands, and the first thing that's going to happen is the pollen grain is going to, as we see, germinate. But what's really happening is it's going to absorb moisture from the top of that stigma. And as it absorbs moisture, it allows that pollen tube cell to elongate and grow down the style. So here is a pollen grain, and the pollen tube cell has already elongated down, and we can see inside the two sperm cells, right? So that pollen grain, the pollen tube cell is going to elongate down the style, and then that generative cell is going to undergo mitosis to produce those two sperm. In the case of angiosperms, the sperms no longer swim. They're non-motile. They don't move. They just move as the pollen tube cell grows. So as the cell elongates, it moves the sperm down. But they don't actually swim anymore. All right, so the pollen tube cell has continued to grow down the style, and then eventually the pollen tube cell is going to reach an ovule. When the pollen tube cell reaches the ovule, it's going to enter the micropyle. Remember the hole at the top of an ovule? Pollen tube cell is going to grow into that, and the two sperm are going to be released. So here they are. There's the pollen tube cell. It has grown through the micropyle. See those two little purple dots? Those are the sperm. There's the egg waiting. And the reason I point out those two polar nuclei is because, remember in the gymnosperms, there were also two sperm cells, but one of the sperm cells would fertilize the egg and the other one would just get discarded. Well, when we get to the angiosperms, discarding anything seems very wasteful. So we try not to discard anything anymore. And so now what's going to happen is one sperm cell is going to fuse with the egg, and that other sperm cell no longer gets discarded. It actually fuses with the two polar nuclei. So I'm going to, so this is an ovule with sperm arriving. One sperm is going to fertilize the egg. One sperm is going to join with the polar nuclei. Once that occurs, boom, you have a seed. So remember the sperm is the purple. So I'll go to the previous. One sperm cell is going to fuse with the egg. The other sperm cell is going to join with the polar nuclei. Boom. Now we have a seed. When the sperm and egg fuse, that's called a zygote, which we're used to. When the sperm fuses with the two polar nuclei, that's called, it creates a thing that's called endosperm, which is food for the developing zygote and embryo, ultimately embryo. And because it's the fusion of two polar nuclei and one sperm cell, all of which are 1N, the result is a 3N or triploid product. Okay. Enough shop talk. Let's see some pretty pictures. Oh, wait, one more slide of shop talk. So this thing we just went through, right? 2 sperm. You have an egg and you have the 2 polar nuclei. 1 sperm fuses with an egg. That's a fertilization event. 1 sperm fuses with the 2 polar nuclei. That's a fertilization event. Because of that, we call that double fertilization. Because you have two fertilization events inside of the female gametophyte plant by the sperm cells. One results in a zygote, which grows into the new sporophyte plant. And the other fertilization event is the endosperm, which is a food source for the zygote. Right, and this is just a reminder of what I just said about there was also 2 sperm during the gymnosperms, but back then only 1 got used and the other degraded. Okay, now let's see some pictures. So, so when you have a flower and fertilization occurs in the ovules, that flower will turn into a fruit, believe it or not. So look here. In A, you have an apple flower, right? You have the Megasporogenesis occur and Microsporogenesis occur, which we just went over. And then the result of Megasporogenesis is a mature ovule, and the result of Microsporogenesis is a pollen grain. You have pollination, pollen grain lands on the stigma. You have double fertilization, which we just went through. And then the result of that is a seed, many ovules, many seeds that are going to be inside of a fruit. So here you go. Look, this is another example in raspberry. In C, we see the beginnings of a flower. In D, the flower is getting more developed. Remember, we're talking perfect flowers here with both male and female reproductive structures. E, we see the final need of the flower. Pollination occurs. Double fertilization occurs. Now the ovules are seeds. And then by the time you get to F, you're already now in a fruit. So we'll watch one of these, but I'll have these located for you on, obviously you'll have access to the slides, but just to give you a visual here instead of just pictures. Yes, the ovary becomes the fruit. Yes, Ashley's asking if the ovary becomes the fruit. Indeed. All right, let's look at this. Let me make it big. Oh my gosh, come on. So this is a pear, so those are pear flowers. And they are going to, oh, you can't see the video. Okay. All right, well, I guess I have to, let me see if I could share it. Share web browser. Hold on. See if this works. Nope. Hold on. Tell me, don't worry, I'm going to post them, but let me just see if you can see this. Do you see anything happening now? Seems to be working. Okay, good. Yes, great. Sorry, I didn't share it before. All right, so here's our, oh gosh, pear flowers. And they have the male and female reproductive structures on them. Pollination occurs. Oh, there's a pear flowers again. Sorry. There you go. So there's the male anthers are purple, and in the middle you can see the female stigma and style. Now the anthers are ready, so they're going to release their pollen grains. And so pollination will occur. Pollen grain will land on stigma. And then there, the flower petals have fallen off, and now the ovary begins to swell because it now contains seeds instead of ovules, and it will continue to swell and swell and swell until ultimately the flower turns into a pear. Okay, cool. Let's see here. Let's see. Hold on. All right. Make that small. So each flower will form a pair. Sorry. Whoops. Oh boy. Each flower will form a pair if it gets pollinated and if it gets fertilized. Yep. Okay, cool. All right, let's go forward. There's also another example here with a pepper. You can watch that whenever you have time. All right, so eventually the seed, which is inside of a fruit, will land on this. So eventually the fruit will either get eaten by an animal, or the fruit will just fall on the ground and start to decompose, and then the seeds will be released. The seed will eventually land on the soil, and then the seed will grow. So here you got, here you go, seed. Here's, oh geez, here, why? No, I don't want to annotate. I don't want to annotate the web. Okay, 1 second, guys. I'm trying to stop. There. Okay. Hold on. I think I have to reshare the. I've never done this when I'm sharing the. Okay, go here. Share this. Okay. All right. Back to this. Yeah. So what I was trying to annotate before is like what I was saying. So eventually the seed. Here's the seed. It falls off, and then the seed will germinate and grow the new sporophyte plant, which it shows above. Oh, yep, see? There you go. So this is just showing you a diagram of the seed emerging from, the seedling emerging from the seed, right? You can see the root tip. Eventually the seed, so here, this is actually a good schematic for lab. I think it is actually in the lab. So this is example, remember we talked last time about eudicots or dicots and monocots. So this is an example in eudicot. Remember, dicot means 2 cotyledons, right? So on the left here, you have the seed, which is already in the ground now. And then you have the new sporophyte plant emerging. First, the roots are going to emerge. Next to that, you see the new sporophyte plant starting to grow, and it's starting to make leaves. But notice that it bears those 2 cotyledons, right? Because it's a dicot. Eventually, those leaves will be large enough, and so the cotyledons is not endosperm. Very good question. So the cotyledons and the endosperm are ultimately the same structure. The difference is, so first, what's the best way to say this? During that double fertilization event, you have the creation of endosperm, which is that 3N food source for the embryo. However, as it feeds the embryo will grow structures, and some of those structures that it grows are these cotyledons, and the cotyledons will take up the space of where that endosperm was. So as the embryo is consuming the endosperm, it's going to grow roots, it's going to grow a shoot tip, it's going to have cotyledons, but the cotyledons will ultimately take up the same space as where that endosperm was. All right, but at this point though, when the seedling is emerging, these are cotyledons. We no longer call them endosperm anymore, right? Because they're not. They're now baby leaves on a plant. Once the plant has these mature leaves, not the baby leaves anymore, the mature leaves, doesn't need the baby leaves anymore, and the baby leaves fall off. Okay, that's in a dicot. Do all angiosperms have fruit? Yes, all angiosperms have fruit. Yeah, so that's a funny question because, we say like fruits versus vegetables. Well, and everyone's like, did you know a tomato is really a fruit? Well, guess what, guys? Did you know a pepper is really a fruit? Anything with seeds inside of it is botanically a fruit. So if it has seeds inside, or is it supposed to have seeds inside, it's a fruit. So the only things that are, in my opinion, vegetables are lettuce or onion or potato, right? So garden bean is a fruit bearing seeds. Yes. Enjoy talking about that with your families. I should love that. Rebecca says that a pepper is a fruit. It is, but it's okay. We still do group them differently. And then this is an example in monocot. I don't really care too much about this, but the point is, remember we talked about that. So 1 dicot in one cotyledon in the monocots, 2 cotyledons in the dicots. How are we doing on time? All right. You guys are used to leaving early, but we're going to probably go the whole way today. Because let's see. Oh, okay, good. Oh, sorry. We're almost done. Oh, I love these. Okay, so let me share. All right, so I'm going to share web browser again. And then I go back here, and then I go, hello, where is it? Go here. All right, so we're going to watch a sunflower plant. Okay. We're going to watch a sunflower plant grow from seed to a plant. If this works. Yep. From seed to flower. Oh, sorry. Don't listen to this guy. I don't endorse him. I don't know what he's trying to sell us. Skip, skip, skip. Don't buy it. Don't buy it. It's a scam. All right. Here we go. So this is, look, there's the cotyledons. Oh, geez. No, thanks. Go away. There's the cotyledons. There are the real mature leaves starting to grow. As they're starting to grow, you can see the cotyledons are starting to shrivel up. Look, now they're all shriveled up, so they're going to fall off. Mature leaves are going to, so look, as this is, what you think about it, what's happening at that apical meristem, right? Apical growth, lengthening, creating all those parts of primary growth, right? Stem, leaves, all the different parts. Okay, now it's going to start to make a flower. Oh, so dramatic. So those green things on the outside of the flower bud are the sepals. And this is when I laugh when people say plants don't move. Plants move a lot. You just have to use time-lapse photography to see it. Now the sepals have started to open. Now the petals are exposed. Look, we can see our composite flower, right? In the disc, we can see the disc flowers. And we see the pretty ray flowers surrounding it. Look, all of the stigmas on the disc flowers are starting to open. So they're exposing, the stigmas are being exposed so that they can be pollinated. As you can see flies flying around here, they are using flies to pollinate these plants. And as pollination occurs, each of those disc flowers will become a sunflower seed. You can see it's starting to turn black. Those are all because they're turning into sunflower seeds. All right, so let's see this next one right here. Oh boy, hold on. 1 second, I'm sorry. Oh my goodness gracious. This is really harder than it looks, guys. Lady, go away. Go away, lady. Stop. Okay. Jeez Louise. I mean, I should have, they didn't trade me for this. Okay, so here you go. This is a kidney bean, another dicot. Dicot, kidney bean is in the ground. The primary root has already come out and is down. Now the secondary roots, our lateral roots are coming out of the primary root. Now the shoot system is starting to grow out. And the seed coat is still covering the seed. It's going to fall off. That's that brown thing. Exposing the cotyledons, right? Those embryonic leaves. Those are at the base right there. Looks like kind of like a peanut. And then as the apical growth is occurring, it's making it's making leaves and it's making shoot system. And then eventually, now that it has mature leaves, eventually the cotyledons are going to fall off. And look at on the left, how much the root system has grown. It started with just, oh, there it went. There was the cotyledons fell off. Yeah, and so then eventually this plant will bear flowers and create more guinea beans. Makes you dizzy, right? Oh my gosh. All right. How much longer we got? We got a little while. Oh yeah, I'll let it go. Fine, whatever. Fine. And you said the beans are a fruit with a seed within. Yeah, so fruits contain seeds. Oh yeah, all right. Well then now it's showing you more about okay. Let's see, is this, all right. Yeah, so you get the idea. So stop sharing that. And let me just also, oops, no, 1 second. Okay, close this. 1 second. Oh gosh, close that. Okay, let me share this PowerPoint slide. So we're almost done. Resume slideshow. Can you guys, oops, where are we? Oh gosh. WebEx. Here we go. Share the PowerPoint slide. Okay. Now you guys should see it. Yeah, so this PowerPoint slide will be available. Whoa. On oh, I saw something pop up. This PowerPoint slides will be so that means the sunflower seed is actually a fruit. Yes, it's true. All right, so we're about to finish up here with thing. So normally in class, I would ask if there's anything regarding angiosperms or any questions about anything that we've covered. If you have any questions right now, I'm happy to answer them, but I'm also happy to meet for office hours, like virtually like this. I'm also happy to answer any emails if you want to ask questions that way as well. If you tuned in late in terms of when is the final test? We're gonna talk about that right now. For the phenology assignment, just give me whatever you got. Follow the instructions, but just send me however many weeks you have of it. If you want to learn more about plant-based careers, not just within academia, this is in a lot of different sectors. This link gives you a really nice overview of all the different types of careers that you can have. And final exam is on the 12th of May. That's the time it's scheduled. It normally is supposed to start at like 8:30 in the morning or something like that. We're just going to start at normal class time, 10 a.m. If you have a final exam conflict, please let me know about it as soon as possible so we can deal with that. And I'll need documentation about that. But if you have a conflict, let me know. So May 12th. I'll post a study guide a week before on the 5th. And where we come to Blackboard and take it here, yes. Yep. Yeah, and hopefully it'll go smoother than the last time. I think now I kind of figured out what went wrong that time. Yeah, so these are, I will post a study guide, but this gives you really what the exam's going to be on, right? It's the evolution of the vascular plants from the spore shedding to the seed shedding. So just understanding how the spore bearing structures have become more complex over time. These are those core course concepts. And understanding how the gametophytes become more reduced over time. So again, I'll post a study guide a week before, and I'm happy to meet you for office hours. So 10 a.m. it's going to be a short exam. It's going to be a short exam. It only covers angiosperms and the core course concepts. It's not cumulative. It is just these core course concepts and angiosperms. I will post the study guide like a week before. Yeah. Let me look at examples of exams I've done in the past, and I'll have a better idea of how much time I think it'll take. And I'll figure it out then. We're gonna end class by watching this last thing. Here you go. Like I said, send me phenology, whatever you have. I think tomorrow is your lab exam. If I'm wrong, don't get mad. I haven't been keeping track of what's going on in lab. It is. I thought so. And then, like I said, I'll upload the study guide. Oh yeah, and there's a course survey, so feel free to fill that out or please do fill that out because I make adjustments every time I teach this course based on what students say. So, the fake test should not be included in the grading. It might show up on the columns, but it's not actually included in the actual grading. I'll double check it obviously before I do anything. Okay. Yeah, it might show up, but it's not set to be calculated towards your course grade. And before I do this little video that we're going to watch, I just wanted to thank you all so much for a wonderful semester and for hanging in there during this crazy time and for dealing with my exhaustion. And I just wish you all the best in anything that you pursue. And like I said, if you ever need help with anything or if I can help you get towards your goals, you let me know and I'm happy to help you with that. Thank you guys. And let's finish with this. And then if anybody wants to thank you so much, guys. And if you ever, like I said, if you want to meet for office hours, I'm happy to. So you just let me know. All right, so let's watch this. It's not too long. And it's a nice way to end the semester. It's got pretty things. Hello. Oh gosh. All right, and it's only 3 minutes long, so skip ads. So I hope you guys enjoy this. That kind of puts the whole thing in perspective. Oh.
You can't tell me stories about butterflies. That's what the story about. I've been filming time-lapse flowers 24 hours a day, 7 days a week, over 35 years. Watch them dance and everything gets tired. Fills me with wonder and it opens my heart. Beauty and seduction, I believe, is nature's tool for survival. And their relationship is a love story. It reminds us they're a part of nature, and we're not separate stuff. When I heard about the vanishing years, we picked up tiny many tribes of believe it's like the canary in a coal mine. If they disappear, so do we. It reminds us that we're a part of nature and we need to take care of it. Motivated me their behavior. Something that I asked my scientific advisor, what motivates pollination? So her answer was more about more. I go, why is that? They say, well, because they want to survive. In order to reproduce. Well, why? And I thought they'd probably say, well, Chip Taylor, our modern plant, nothing lasts forever. Everything in the universe wears out. And that's what's going on. And I realized, and then reproduction. Rarely seen by the naked eye. So here's the message I hope you'll break the presence of life story. Don't take
Agent of pollination. Oh no. Oh, fighting. I think so. Yeah, they were fighting. So these are, look, these are all nice, great examples of biotic agents of pollination, right? Oh, here comes a bee. That's a bee. Sweat bees. And now some, I think those are, yeah, those are still sweat bees, I think. Look, so look at all those pollen grains flying all over the bee. I know. It's okay. So look, so the bee, they landed, pollination occurred, double fertilization occurred, and now these strawberry flowers grow into strawberry fruits. There's your beans you keep asking me about, your peas. There's a cactus flower, which are usually bat pollinated. They're feasting. Look, the bat's carrying a baby. So this is a forest in Mexico where my monarch butterflies migrate to every year to mate. And when they're there, the trees are so heavy with butterflies that the trees actually sag with the weight of the monarch butterflies. Oh, it was 7 minutes long. All right, guys. Well, it's been a great semester. I wish you all the best. Like I said, I'm here for you. And go out there and be awesome and chase those dreams. And like I said, if I can help you chase them, let me know, okay? Thank you so much. If you want to meet with office hours, you let me know, okay? Take care, guys. Thank you guys so much. Thank you, guys. See you guys again. See you again.